So the study that I presented at AACR described the discovery of a novel small molecule that we've called PC14586 that was designed to reactivate a particular hotspot mutant of p53 called Y220C. The work was performed at the company that I work for, PMV Pharmaceuticals, and there we are a precision oncology company pioneering the discovery and development of small molecules that selectively reactivate p53 hotspot mutants. So p53 is an important protein in cancer, with over 50% of all human cancers having a mutation that renders the protein inactive and non-functional. p53 is a classic tumor suppressor protein, and when it's mutant, it's really like taking the brakes off cancer cell division. So to date, there's not been a therapy to directly treat patients who have these mutations, and that's what the talk was about. With the Y220C mutation, which the molecule targets, that substitution of a tyrosine for a cysteine at the 220 amino acid of p53 results in a small crevice forming in the protein. And the chemists at PMV have designed a small molecule to fit in that crevice and stabilize the mutant protein into a normal conformation. And then we go on to show that the small molecule engages with the target, the Y220C target selectively, meaning it doesn't bind or activate normal wild type p53, which spares normal tissues. And it results in turning on the p53 transcriptional pathway that ultimately results in cell cycle arrest and cell death. So in short, we showed that the chemical reactivation of the mutant Y220C p53 really did does walk and talk like a normal or a wild type p53. And then finally concluded my talk like telling folks that how excited we are that this new investigational compound PC14586 is in clinical trials started last September, and the clinical study is a seamless phase 1-2 study in cancer patients with a variety of tumor types, but their tumors must have that Y220C p53 mutation. Yeah, so I think it was a very kind of logical drug development approach to finding and characterizing this molecule. I think the biology that we showed was very specific to p53 biology, but unique, because I think it was really the first time that anyone had shown with a chemical stabilization of the mutant to the wild type, what happens in the cells. And we really ask at our company that our chemically restored mutant p53 really does look like the wild type protein. So methodology wise, the chemists used a very standard SAR, or a structure activity relationship medicinal chemistry approach, where they iteratively made molecules, and the biologists at PMV test these molecules through a sequence of assays to find molecules that really did have the desired p53 biology as well as potency. We always ask that our molecules chemically reactivate that Y220C mutant p53 and have it look like normal or wild type p53. And that by that I mean that we can show that we can convert the conformation from looking like wild type to looking like mutant to looking like wild type, and that wild type can then bind to DNA. It can activate a p53 transcriptional signature, and then that can result in a very p53 dependent cell cycle arrest and apoptosis. We then optimize the molecules also to have robust pharmaceutical properties, which means that we can administer them orally to either animals and, of course, patients once we make it to the clinic, and show a robust dose-responsive anti-tumor effect. And we showed that in the study, that at the maximal dose, this molecule results in almost complete tumor regression, sometimes complete cure in mouse models of cancer. I mean, I think the takeaway from the study was that we really did describe a first-in-class reactivator of Y220C mutant p53 that is in clinical trials and is available to patients. The molecule is selective for Y220C mutant p53. It binds the target and it reactivates normal p53 function. This results in robust single-agent anti-tumour effect, which increases with dose, so is dose responsive. And we've also established several biomarkers preclinically that we are deploying in our clinical study. And I think for the clinicians that see this, I think it's important to note that the clinical study is ongoing, and it's a seamless phase 1/2 study that is targeting patients who specifically their tumours harbour that Y220C p53 mutation. We expect to be able to report on the safety, PK and biomarkers from this study by the end of this year, early next year. Yeah, so to date, there is no marketed therapeutic agent, nor anything that we're aware of that's currently in development for patients that have p53 mutant tumours. This molecule really would be the first ever reactivator of a hotspot mutant p53. So for patients who have this Y220C p53 mutation, this really is a custom-made therapeutic option for them. And we really hope to be able to improve the outcome for these specific patients. I think more broadly, this work is a solid proof of concept that targeting hotspot mutant p53 is a valid therapeutic approach. So we're of course actively working on other programs to address other hotspot mutants. Our goal is to have custom therapies for the majority of patients whose tumours have a p53 mutation. And then with regards to cancer research more broadly, I mean, we hope that this encourages other groups to tackle other undruggable cancer targets, such as p53, which will really add to the arsenal of cancer therapeutics and provide a wider therapeutic option for patients who are in need. I suppose one thing we didn't touch upon is the frequency of Y220C mutation in tumours, and then how would we use this clinically. So the Y220C mutation is found in about 1% of human cancers, and it's found across tumour types broadly. So it's not enriched in a particular tumour type. So the tumour types where we think we might expect to see the highest number of patients include ovarian, breast, non-small cell lung, colorectal, gastric esophageal, some of those that have, you know, a large number of patients each year. But of course, you know, we may see tumours from many different tissues on our clinical trial. So our clinical trial is recruiting patients with any solid tumour type. So it's what we call a tumour agnostic trial. So long as the tumour harbours a Y220C p53 mutation, and we're using a couple of different sequencing tests to confirm that patients are the tumours do contain the Y220C mutation. As I mentioned, the study, it's probably worth mentioning again, the study is a seamless phase 1/2 study, and we expect to be able to report on the safety, PK and biomarkers by end of the year, early next year.